for coming here today. Thanks a lot for holding with all these technical problems. Now we're going to go from like the highest of the high tech to a little bit of something a little bit more simple. As I mentioned before, just some minutes ago, I'm a biologist, but I think by the end of this, you may be thinking, given the amount of clay I'm going to use, that maybe I'm a kindergarten teacher. Anyway, as I was mentioning, biologists, we aim to study the processes of life. And most, if not all, of these processes happen at a really, really, really small scale. In fact, most of this happens inside the cells. In your body, you have millions of cells of thousands of different cell types. You may have cells of your skin, cells of your brain, and this little fella over here that looks like a fried egg, this is what we call a stem cell, which is capable of becoming any of these kinds of thousands of cells. They have different shapes, they have different functions, they have different behaviors, but all of them have one thing in common, aside from my bad ability of drawing these things, which is that all of them have this red thing, which is what we call a nucleus. Now, inside of this nucleus is the DNA, is your genetic code, is all the information, the most important manual of instructions that you're going to have in your life, which makes you, you. Now, most of you may have been used to see these manuals of instructions as something like this. G's, T's, C's, A's, and things that go for pages and pages and pages. And this is the way we choose to represent it, because it's true, it's a lot of information, but it may be obvious for you that you don't have tiny little sheets of G, T, C, and other letters inside of your nucleus that tell you how you have to be you. What you have is the molecule of DNA. Now, the molecule of DNA is one of the most complex biomolecules that exist, able of amazing things and very, very, very complex. But for today, we have to think of it as a thread. I hope you can see this through Zoom, but I'm now holding one thread of around 2 meters. Now, inside of every one of the millions of cells that are inside your body, you have 2 meters of DNA, not in total, inside of every one of the single cells. You have this. And you have to imagine that you have to pack this in a really, 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 really small space. So now, we have a saying in Spain that says that something can be more untidy than a tailor's cupboard, which if you have ever lost your headphones or put like some cables in a cupboard, you will notice how things begin to tangle and mix, and then it's impossible. You wouldn't want this to happen to your DNA, right? The cell needs to have a way of organizing this. So how do we do it? In big life, I'm not going to say real life because at the cells is also real life, we use what we call bobbins. This, we can coil the thread around it. And it's really practical and it's really convenient. But you have to think that if this was the case, you would have access to this information here. But we wouldn't have a way of having access to the information inside. Does that mean that the cell doesn't use bobbins, that the cell doesn't use this? Not exactly. Come down with me a little bit. This is, well, this is a ball of clay, obviously, but this represents an histone. This is one of the millions of molecules that you have inside of every nucleus in your body, which helps it organize the DNA. But this small histone doesn't work alone. It works in 8. This is what we call an histone octamer. 8 pieces of 4 different histones. And if you remember the bobbin from before, you can begin to see how things are a little bit similar. So now, inside of the cell, we have DNA, which is wrapped approximately twice around the histone core. This is called the nucleosome. And this is the basic unit that forms the structure inside of the nucleus. Now, this has already been known for quite some time, but what we still don't know and what we are doing in our research is understanding how the histones are going to be distributed in the physical space between them and how this is going to affect the function. But this is really, 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 really small. So as I mentioned before, we need to use super resolution microscopy because otherwise we won't be able to see. So now I'm going to show you the first ever full cardboard microscope, super resolution microscope. So what we are going to do is we're going to attach things that blink to what we want, the structure we want to see. And we're going to take a really long movie. We're going to stop for a moment one of the frames of this movie, and every frame of the movie will have these blinks, and in this frame we'll have this, and this. These are single molecules, and we can find their localization with the highest precision today with optical microscopy. And we can pinpoint exactly where these DNA and histone molecules are. So now let's imagine for a moment that we repeat this process over time, over thousands of frames, over millions of molecules, and at the end, we'll have the structure that we want. Now, the obvious thing that you're asking is, what does happen with the histones then? Well, now, first of all, we have to minimize things a little bit, simplify. So we are going to go from this to this. I want to imagine this as the Octamer histone that I showed you before, with the DNA wrapped approximately twice around it. So this is more or less the structure that we knew. And when we apply this microscopy, I just showed you before, we have finally been able to reveal what is the structure of the chromatin. Now, this is a representation of one of the latest information known about how the chromatin is structured. And one of the most surprising things is that it's not a regular structure. It's not all organized in the same way. You have regions, small regions, in which like the DNA is compacted together with the histones, forming what we call histone clusters, compacted clusters. These places is where the information that we don't want the cell to read is stored. And there's other more open regions, which are easy to access, easy to read, easy to interpret these instructions. The amazing thing, actually the amazing thing is that it's not like things can be open and closed. There's like intermediate states. It can be closed, but like in a smaller cluster or an even big cluster than this. And the most second amazing thing is that this is not always to be like this. Like this cluster can become an open or can close depending on the cell type, depending on what is the function of the cell. This can become like this, or this can become like this, and vice versa. So you may be asking, why is this important? Why do we need to know that? Understanding the way the nucleus is structured, finding which are the players that do that, and achieving and resolving which is the mechanism that alter this and that produce this structure, allow us to understand processes as important and as different as cell differentiation from embryos to adults, such as cell death, but also pathological processes such as cancer, in which this structure becomes absolutely changed. This is one of the most fascinating for me questions of biology, and the one we strive to answer. Now, Aldous Huxley, one British writer, once said, words are the threads that we coil around our experiences. Well, what we say is that DNA is the thread that we coil around our being. Thanks a lot, and I hope you enjoyed it.